Hello everyone, welcome to PW English YouTube channel. I'm Shalini Somashekhar, your botany teacher. I welcome you to today's lecture in the UMEED NEET English batch. In today's class, we are going to be discussing the chapter Cell Cycle and Cell Division in one shot. But before we start with the discussion, let's understand the weightage of this chapter and how many questions can be expected from this chapter. You're always worried about whether you should refer old NCERT or new NCERT. For this chapter specifically, either of them works. Last year, I think after the NCERT got rationalized, some concepts from some chapters were deleted, so that confusion was there. But this chapter, even after rationalization last year, was untouched. All the concepts that was there in the old NCERT were still retained in the new NCERT textbook, so you can study from either of those. I'm sure all of you must have gone through the new NMC syllabus that has been released for 2025. It is exactly the same as it was for NEET 2024. So there's no change in the syllabus, and here is a snip of the syllabus for cell cycle and cell division. We have to study about cell division, of course, and then what is a cell cycle, what are the different phases in a cell cycle, what are the two types of division, mitosis and meiosis, and what is their significance? How exactly does it occur and what is their significance? That's what we'll have to learn. All right. Now, talking about the weightage, you can expect anywhere between 4 to 6 questions from this chapter. If you look at the past few years, just in the last NEET exam 2024, 4 questions have been asked. Before that, 6, 5, 5, 4. So consistently from 2020, you can see how the number of questions have been ranging between 4 and 6. So that's how much you can expect from this chapter in the upcoming NEET exam as well. Another thing, though we generally teach this chapter under botany, questions from this chapter in the actual NEET examination can be asked both in botany and zoology sections. It's usually like 3, 3, 2, 3. In both the sections, you will find questions from this chapter. All right? Yeah. So let's get started. What is the cell cycle? The chapter is called Cell Cycle and Cell Division. We are of course going to learn about cell cycle and how cell division occurs here. And this chapter, you will learn about a lot of different phases in the cell cycle, what event takes place during which phase. Questions are mostly asked on that. So they will be memory based and you have to pay attention, understand everything really well, revise frequently and remember the events properly. Then this chapter's questions is going to be a walk in the park for you all. Alright, so let's get started. You know that all living organisms, there are certain characteristic features that they exhibit. When we started off with this batch, we started with the botany chapter, the living world. I explained to you what are the different characteristics of life. Some of those characteristics are growth and reproduction. This is exhibited by all living organisms. Right? Now, living organisms can be of two types. You have unicellular organisms like bacteria and algae, and you have multicellular organisms like us, right? Unicellular organisms are made up of just one cell. Multicellular organisms have very many cells. Basically, they have more than one cell. Right? Multicellular organisms are made up of more than one cell. Let's first consider the case of unicellular organisms. What happens in unicellular organisms like amoeba, let's say for example? They exhibit growth, they exhibit reproduction as well. So if you have like one amoeba cell, this amoeba cell as it grows increases in size and then eventually The growth in unicellular organisms is equivalent to reproduction also. They are synonymously used. We've learnt about it in an earlier chapter. So here, when a euglena or an amoeba grows, the amoeba that I've drawn here, when it grows, it can grow only up till an extent. This unicellular organism cannot grow as big as a human. It cannot grow as tall as me, right? So after one point, it will undergo cell division and it will give rise to two small amoebae. Again, the process will continue in those amoebae. They will grow in size and they will divide. So when growth is happening in unicellular organisms, there is increase in size, of course, to an extent, but after that there is increase in number, which is essentially reproduction, right? Reproduction here is because of this cell division is happening. So growth and reproduction both can be used interchangeably or synonymously in case of unicellular organisms. Now, when it comes to multicellular organisms, especially when you consider sexually reproducing multicellular organisms like us and many organisms, if they are sexually reproducing, how do they form? By the formation and fusion of gametes. The male gamete, sperm, in case of animals, otherwise the male gamete in plants, the male gamete, for example, I'm taking the sperm, and the female gamete, which is the egg cell, they fuse by a process called fertilization and form a zygote that is diploid, right? This will be diploid, this will be haploid, and this is haploid. So essentially, we all sexually reproducing multicellular organisms started our life as a single cell. Right now, I might have trillion cells in my body. Right now, a dog might have trillions of cells in its body. Right now, as adults, we might be multicellular. But how did we start our life? Our life started as a single cell. And how did that one single cell become trillions of cells? One cell divided to give rise to two cells divided to give rise to 4 cells, then to 8 cells, then to 32 cells, and so on. Along with cell division, differentiation also took place inside the womb of the mother in case of us, and then eventually the baby is formed, it's mature, and then the childbirth happens, right? So regardless of whether you're a multicellular plant, multicellular fungus, multicellular animal, regardless of what it is, if you are a sexually reproducing multicellular organism, then essentially our life is going to start as a single cell, which is the zygote. So cell division is very important because it is because of cell division that one cell became like trillions of cells, right? It is because of cell division that a unicellular organism became multicellular. So that is very important. Also, there are many other significance of cell division about which we'll learn moving on. Okay. Now, what is a cell cycle? You know, similar to how we have our life cycle. When does our life cycle begin? Our life cycle begins the moment we are born, right? Or you can even say the moment we are formed as a zygote. That is when our life cycle begins. Let's consider birth for convenience. So we are born, our life begins on earth. And then there are so many phases in our life. The starting point is birth, the end point is death. In between, you have so many different phases, right? Initially, there is infancy. The small baby is there, there's an infant. That's infancy. After that, you have childhood or toddlerhood. Then after that, you will have childhood, of course, and then you'll have adolescence, and then you'll have like adult. You'll become a young adult, and then you'll be middle-aged, and then you'll be aged, and then we die, right? We age and then we die. So we have very many phases in our lives. Similar to how we have a life cycle, cells have their own life cycle. The life cycle of a cell is called its cell cycle. Similar to how our life has many phases, a cell's life also has many phases. All of those phases, all of those events that are taking place in those phases is what we're going to learn about now. Okay. So similar to how in each phase there is some distinct characteristic events that take place, right? So when we are born, initially Babies, when they're born, they can't see color vision. At one point, they will develop color vision, probably around 5 months of age. And then they will start feeding on solid food. And then probably they'll start walking. So you have events that happen in different phases. Around adolescence, they will hit puberty. Around old age, their organs will start deteriorating. They will not function as efficiently. So during different phases, different events will take place. Very similarly, even in the cell cycle, in the different phases, different events will take place. I'll give you an overview of what all events could take place, will take place in a cell cycle. So you get an idea, and then we'll get into the details of it. Okay. So let's start with a baby cell. It's just now formed, super tiny, cute little baby cell. It's very small and it's formed right now. Let's say this is a diploid cell. What happens when a cell forms is it will be very small. As it grows in size, it will accumulate more and more cytoplasm within. It will start becoming bigger. Okay, it's still diploid. So the cell will undergo growth. Growth is a very important part of the cell cycle. Small cells will increase in size by accumulating more cytoplasm. The volume of cytoplasm will increase. Cytoplasmic volume is going to increase. Cytoplasmic volume is going to increase. But let's look at the nucleus. Here you have a diploid nucleus. Here again you have a diploid nucleus. This is the nucleus. The nucleus is unchanged even after the cell has increased in volume. The cell has grown bigger, but the nucleus remains as it is. Why is nucleus important here? Nucleus contains our DNA, which is the genetic material. Genetic material has information for pretty much everything that's taking place inside the cell. Okay. Right now, the cytoplasm volume has increased, but the DNA content has not increased. Nucleus can be thought of as the boss of the cell. Now that the nuclear content is the same, but the cytoplasmic content has increased so much, it becomes difficult for the nucleus to manage all of the activities inside the cell. When that happens, we say that the nuclear cytoplasmic ratio is disturbed. Nuclear cytoplasmic ratio is disturbed. Okay? Now think about it like this. If you have a teacher in your class and in that class there are like 40-50 students, it's easier for the teacher to manage the class. By looking at the faces of the students, the teacher can pretty much say who is understood, who hasn't understood, who is looking confused, is everybody listening, is somebody sleeping. She has a clear idea of what is happening in the class, she or he, whoever the teacher is. Now imagine there's one teacher who has to manage a class of 1000 students. It would humanly not be possible for the teacher to give individual attention to everybody in the class. There could be somebody who's listening to music, there could be somebody who's just playing mobile games, there could be somebody who's just dozed off. So the teacher will not know. Same thing happens inside the cell. The nucleus, the same amount of nucleus in this whole volume of cytoplasm will not be able to very well, control the activities. Another thing is, if any nutrients or any substances have to be diffused inside the cell. Let's take for example, there is a blood capillary. Blood capillaries carry oxygen and nutrients to every one of our living tissues and supply it to our cells. When the volume of the cell becomes really big, the nutrients or oxygen or whatever, if it has to diffuse inside the cell, and if any substance within the cell has to diffuse inside, the distance will become very longer, which is not a very great thing for the efficiency of the cell. Therefore, when this happens, when the nuclear cytoplasmic ratio gets disturbed, the cell decides, I can't manage all of this right now. It's becoming difficult for me, so let me divide. It's like there was one teacher who could comfortably handle a class of 50 students, then suddenly you gave them like 1,000 students. It's becoming difficult to manage. How can you better manage this situation? How do you tackle this situation? Ideally, if a teacher can manage 50 students effectively, bring more teachers, divide the class, split them into groups, and give 50 students to each teacher so that all the students are taken care of. Alright, very similarly, even here, the cell will decide to divide so that when the cytoplasmic volume comes down, it's easier for the nucleus to manage it. Alright, so when this happens, when the nuclear cytoplasmic ratio is disturbed, that is when the cell makes a very important decision to divide. Because of this same reason of diffusion diffusion having to happen over a long distance and nucleus unable to control all of the activities, that is why you don't see unicellular organisms that are huge. You see unicellular organisms that are super tiny, they don't grow much, because if it's unicellular and uninucleate, it cannot function properly. Alright? Yeah. So then it decides to divide. Alright, it has decided to divide right now. Let's take this cell here. I'm going to draw it here again. This is one big cell right now. Within it, I have a nucleus. And in this nucleus, there is DNA. In different segments of the DNA, there will be information for different things. In this one segment, let's say, okay, in this segment, there is information for hair texture. In this segment, there is information for probably a protein that is required in ATP synthesis. Over here, there is an information for probably a molecule that is involved in cellular respiration. Okay. Now, when the cell divides, what's going to happen? When the cell divides, you're going to get two smaller cells from one big cell. Obviously, the cytoplasm volume is going to get reduced if one cell is going to divide into two. But what about the nucleus? Nucleus has DNA which has genetic information. If I randomly cut this nucleus like this, let's say, and one part of it goes into this cell, the other part goes into this cell, what would happen? Both the cells will get only half of the information or partial information. Complete information is not there. This first cell will not have information for some protein required in cellular respiration. This cell will not receive information for probably ATP synthesis and a protein required that controls hair texture. So, incomplete information will be passed on to the next cells. If these decide to divide again incomplete information will be passed on. For a cell to function normally, the complete set of information has to be there. So, before the cell undergoes cell division, the cell has to make an extra copy of its genetic material. Extra copy such that there are two complete copies and one copy is passed on to each of the daughter cells. Okay, think about it like that. One of your seniors or one of your teachers have like one super study material which is handwritten and it has all the mind maps and everything that you need to kind of revise for NEET exam. And you want, that person, your senior, has obviously done really well in NEET and he's studying MBBS right now, and you want that notes. Your friend also wants that notes. So both of you go ask your senior and your senior very graciously gives it to you. What will you do with that notes? Let's say in one half, the first half of the book, there is information for first year 11th grade chapters, and the second half there is information for 12th grade chapters. Will you like cut it in half and one person takes one part and the other person takes the other part? Will that be a wise thing to do? If at all you do that, then the person who took the second part will not have any information on the first 11th grade chapters. The person who took the first part will not have any information for the 12th grade chapters. So the wiser thing to do is to go and get this photocopied so that you have two complete copies, and whatever is the cost, you both split it, right? So that is what the cell does too. The cell, before it divides its cytoplasm, will make sure and very carefully make an extra copy of its DNA. Okay, so a DNA copy is made. An extra copy is made. This process is called DNA replication. Replica means exact copy. DNA replication means DNA is making an extra copy of itself which is exact. Okay, whatever was the information for hair texture, probably wavy, ATP synthesis, cellular respiration, whatever information was there here, the exact copy of that will be made in your DNA. Okay, so that is called DNA replication. That process is called DNA replication. More about how exactly it happens and all of that we'll learn in another chapter called molecular basis of inheritance much later. For now, just remember, an extra copy of DNA is made. So after this extra copy of DNA is made, the cell will divide. And this process is called cell division. So in the life of a cell, three very important processes are going to take place. Firstly, cell is going to grow. Secondly, it's going to replicate. Thirdly, it's going to divide. Cell division. Before cell division, the cell has to grow and it has to make a copy of its DNA. Now, this cell growth is a continuous process. This growth is a continuous process and replication happens at a specific phase in the cell cycle. Okay, which is that phase? We'll learn about it. For now, I hope you've understood what is the need for a cell to divide. Why can't it be huge, the size of us? Because it's difficult for one nucleus to manage all of the activities. That's why it's going to divide. But before it divides, it has to ensure that both the daughter cells will get the exact same amount of DNA which has all of the information. Therefore, it undergoes replication and then it will divide. Okay? Yeah. So, what is a cell cycle? How can we define it? A cell cycle can be defined as the sequence of events. It happens in an orderly manner. Okay? So, it is a sequence of events by which the cell duplicates its genome, which is basically DNA replication, synthesizes whatever is required for cell division, and eventually it will undergo cell division to give rise to daughter cells. This whole sequence of events is what we call the cell cycle. Why are we calling it a cycle? Because it is what will continue and repeatedly happen. So one cell was small baby cell, it grows in size, it replicates its DNA, divides, gives rise to two small cells. These two small cells will further do the same thing, right? So yeah, all of these events are under genetic control. Okay, so the DNA controls when, what is happening, how it's happening, and all of that. All right, now let's talk about the different phases in the cell cycle. Similar to how I've already mentioned earlier, like how our life cycle has so many different phases, the cell cycle also has many phases. Okay, we'll learn about it. So, when you consider a typical eukaryotic cell, for example, some type of human cell which is showing cell division, if you're growing it in a laboratory, if you're growing it on a nutrient medium, basically if you're culturing some sort of human cell which is dividing, we notice that once in every 24 hours, it will show cell division, approximate number. It's not like today divided at 10 a.m. and then tomorrow morning sharp at 10 a.m. it's going to divide. It doesn't work like that. Approximately every 24 hours, on an average, it's going to show cell division. Now, this duration is not the same for all types of eukaryotic cells. Some eukaryotic cells like yeast divide once in every 1.5 hours or 90 minutes. And if you consider one organism, a human alone, within a human you have so many different types of tissues, right? Even within a human, different types of tissues, different types of cells divide at their own rate. So the 24-hour thing is an average for typical human cell that is grown in culture. In our body, some cells might divide more frequently, some cells might divide less frequently. So that's just an average number. So it varies from one organism to another, and even within the organism, it will vary from cell type to cell type. So this duration of a cell cycle is variable. Okay, talking about the different phases, there are two major phases. The first one is the interphase and the second one is M phase. M phase is where actual division happens. Interphase is, inter means in between. Interphase is what happens, or the phase between two consecutive cell divisions. Okay, so if you think of this as the cell cycle, from the time the cell is, brand new cell is formed here, to the time it increases in size, and here in the M phase it undergoes division, till then this whole thing is the cell cycle. So within the cell cycle you have two distinct phases. The first one is interphase, like I already mentioned. Whatever I am outlining in pink, this whole portion of the cell cycle that I have outlined in pink, that is our interphase. As you can see clearly from this illustration, interphase occupies a lot more time. The cell is present in interphase a lot more time than it is present in division phase. So this here is the M phase. This is M phase. M phase is the phase where the cell is actually dividing. Within M phase, mitosis or meiosis, either types of cell division can take place. So in a cell cycle, cell division is one small part. Before the cell undergoes cell division, it has to grow in size, it has to replicate its DNA, it has to make a lot of necessary arrangements. All of that happens in interphase, where the cell is preparing to undergo cell division, where it is growing and increasing the cytoplasmic volume. So interphase takes a lot more time compared to the actual phase where cell division occurs. Okay, so what is M phase? M phase is where the actual division or mitosis takes place. What is interphase? Inter means in between two successive M phases. So here one cell is just now formed after a cell division. Now this cell will grow, this cell will undergo cell division. So from one cell division to the next cell division, whatever is there in the life cycle of a cell, that is its interphase. Between two consecutive cell divisions, whatever occurs, that happens in the interphase. So I told you that on an average, human cell divides once in 24 hours. If you take that example, in human cell, it's dividing once in 24 hours, means the cell cycle is lasting for 24 hours. Among those 24 hours, the cell is actually dividing in less than 1 hour. The remaining 95% of the time, the cell is in interphase itself. Okay. Now, within the interphase, you further have three phases, G1, S and G2. I will highlight only the interphase for now, and then we'll study about the M phase later. So this is the interphase, right? The cell cycle is proceeding in clockwise direction like this. The first phase after division in interphase is called the G1 phase. After the G1 phase, you have the S phase. After the S phase, you have the G2 phase. So in order, it is G1 followed by S followed by G2. Okay. So interphase, generally it is also called resting phase. But the cell isn't really resting here. It's doing a lot of things. It's performing a lot of metabolic activities. But we say it's resting because it's not like dividing at that moment actively. That's why. But the cell is not like lazy and not doing anything. It's growing, it's replicating DNA, it's doing a lot of things, but it's not dividing, right? So the cell during this phase prepares to undergo cell division later. During the interphase, two important steps happen. I've told you this earlier also. In cell cycle, you have three steps: growth, replication and cell division. Cell division happens in the M phase. Okay. Cell division happens in the M phase, and during interphase, the other two processes, cell growth and DNA replication, will take place. Now, cell growth, like I mentioned earlier, is a continuous process that happens from G1 through S through G2 as well. But DNA replication is a very, very specific process that happens specifically at S phase. S for synthesis, S for DNA synthesis, that's why it's called the S phase. Okay. So, the three phases are G1 phase, S phase and G2 phase. What is G1? Gap 1. G2 is gap 2, because this gap phase happens after mitosis. You also call this post mitotic gap phase, because G2 phase happens before. Remember, this is proceeding in clockwise direction. This comes after this comes before this. So G2 is coming before the M phase. So G2 is also called, pre means before, right? So pre-mitotic gap phase. Now we'll individually learn about what happens during G1 phase, what happens during S phase, G2 phase, all of that. Starting with G1 phase, this is an interval. This is our G1 phase. Whatever I have outlined here is the G1 phase. Where does it come? It comes in between M phase and S phase, where DNA replication happens. So it is a phase that happens between mitosis here and initiation of DNA replication in the S phase. Here the cells are basically metabolically very active. They are growing, increasing the cytoplasmic volume. Remember, in G1 phase, replication of DNA does not happen. It does not happen. Okay, in the G1 phase, it does not happen. It happens in interphase, but specifically in the S phase. That's why it's even called the S phase. Okay, now talking about S phase is where replication of DNA will take place. You call that DNA synthesis. The S in synthesis is what this S represents. So here you have the S phase. During S phase, replication of DNA, making an extra copy of DNA will take place. Whatever enzymes are required for DNA replication, whatever else is required for DNA replication, all of those preparations would have happened prior in G1 phase itself. So at S phase, the cell is perfectly ready to replicate its DNA, and it does the DNA replication. Now we must understand one thing here. Okay, a lot of students get confused at this point. So I will explain this to you. Let's take one cell. Okay, this cell is diploid. I'm showing you just the number of chromosomes inside. I will not mention the other details of the cell because that is not important right now. Let's say in diploid condition, this cell has 4 chromosomes. Okay, each chromosome will have 1 chromatid. This is when the cell is in G1 phase. When it is going through S phase, in S phase, DNA replication will happen. The DNA content will double. Diploid cell will not become tetraploid, it will remain diploid. But there will still be 4 chromosomes, but each chromosome now will have an extra chromatid. So whatever was here, it got copied. Whatever was here, it got copied. Now you still have a diploid cell. Diploid did not become tetraploid after DNA replication, right? Here also you still have 4 chromosomes. But over here each chromosome will have 2 chromatids. This is at the end of S phase after DNA replication has already taken place. Now this condition where you have a diploid set of chromosomes, 2 sets of chromosomes, you call this 2C condition. When replication happens, the DNA content doubles, chromosomes don't double. So this is called 4C condition. Okay? So after replication, remember very clearly the number of chromosomes don't increase, the DNA content only doubles. What would happen after mitosis? During mitosis, the centromere, you know what a centromere is because we've already done that in another chapter, previous chapter. It will split and you get 2 cells. Eventually, each cell will have the same number of chromosomes, same number of chromatids, same number of DNA content as the previous cell, as the parent cell. After cell division, you will get this, where each cell is diploid and back to 2C condition. Okay? So when replication happens, initial amount of DNA is denoted as 2C. After replication, after the chromosome makes an extra copy, an extra chromatid is formed. After DNA content doubles, we call it 4C condition. Remember very, very clearly, there is no increase in the chromosome number. Okay? Chromosome number will not increase. You will get application based questions like, okay, at this point in this cell, the diploid chromosome number was this, how much will it be after S phase? Questions like that. So always remember the number of chromatids will be double, but the number of chromosomes will not change. How do we count the number of chromosomes? We count the number of chromosomes by counting the number of centromeres. Over here we have 4 centromeres, so that's equivalent to 4 chromosomes. Here also you have 4 centromeres, which is equivalent to 4 chromosomes. Okay? Yeah. So there is no increase in the chromosome number. You know that animal cells have an organelle called centrosome, which will have 2 centrioles perpendicular to each other. We've learnt about it in our previous chapter. So this centriole, which will assemble all the spindle in animal cells during mitosis, that centriole, which is involved in spindle formation, later will also make an extra copy. Normally, one cell will have one nucleus. Close to the nucleus, will have one centrosome with 2 centrioles. But before cell division, that also undergoes replication. So this, remember, centrioles duplicating, this happens in animal cells only. This also happens during S phase. So in S phase, 2 things are getting synthesized. One thing is DNA. This is in plants, animals, microbes, every eukaryote. And in animal cells specifically, centrioles will also get duplicated. Okay? Yeah, that's about the S phase. Now let's talk about the G2 phase. Here is our G2 phase. During G2 phase, you can see how whatever proteins are required for what is coming next, M phase, where mitosis or meiosis will happen. For that, whatever requirements are there, those preparations are made in the G2 phase and the cell basically continues to grow even over here. Proteins are synthesized in preparation for mitosis. Remember, I told you that growth is a continuous process. It doesn't happen in G1 phase and stop at S phase and then stop after G1 phase. So it's a continuous process. The cell will keep increasing in size throughout the interphase. Okay, so even in G2 phase, the growth will still continue. Now there's another phase called the G0 phase. Now you might wonder that in humans, there are some cells that don't divide very frequently, like the cells in our heart muscles, our neurons, they don't divide very frequently. If a cell is not dividing, then which phase is it in? Will it be in G1 phase, S phase? What exactly is that cell doing? Which phase it is in? Some cells that don't frequently undergo cell division, which don't undergo cell division, they may not undergo cell division further at all, or unless necessary, unless called upon, they will not undergo cell division. Such cells, they are said to exist in a phase called G0 phase. G subscript 0, it's called G0 phase. Okay, so here some cells do not appear to exhibit cell division, such as our heart cells. They or some other cells that divide only when it's necessary, very rarely, then they exist in this G0 phase. So what happens in such cells is, since they are not dividing, they know they are not going to divide for a while unless it's absolutely required. For example, say there is some injury or something and then cell division might be required. Unless that is required to divide, it will not undergo cell division. If it doesn't undergo cell division, should it really replicate its DNA? Replicating DNA is not essential. So such cells, they do not grow so much that DNA replication has to happen and the cell has to divide. They will increase in size to a considerable amount in G1 phase, and after that they will stop further increasing in size to a point where the nuclear cytoplasmic ratio will get disturbed. Okay, only if that happens the cell will divide. If they are not growing to that extent and growing only a little bit and stopping their growth, then there is no need for further phases, right? So these cells are said to exit the cell cycle. They leave the cell cycle at G1 phase and exist in a quiescent stage called the G0 phase. Okay? Just because it's in the G0 phase, we cannot say that the cell is not metabolically active. It's doing all the functions that a normal dividing cell is also doing. But why are we saying quiescent, which means inactive or dormant? It's because it's not actively dividing. It stopped dividing because it's not required by the body or the organism right now. And it has grown a little bit in size, not to a point where the nuclear cytoplasmic ratio is disturbed, and then it leaves. And it stays there in G0 phase until it's called upon to divide. Okay? So, such cells exit the G1 phase. They exit at which point? They exit the G1 phase to enter an inactive. See, we are saying it's an inactive stage. Inactive with respect to cell division. But they will still be metabolically active. Okay. So, they will no longer divide. Proliferate is the word used in NCERT. They will no longer proliferate, divide and give rise to more cells. But they are metabolically active. If, let's say by chance, that cell is required to divide again, what will happen? Wherever it exited, from the same point it will enter, it will come back to G1 phase, prepare whatever is required for DNA replication, replicate DNA, prepare for cell division, go and divide. That is what it will do. Okay, depends on the requirement. Now, let's talk about the actual M phase or mitosis phase. Though we say M phase is mitosis phase, in M phase meiosis also can occur. In animals, mitosis generally occurs in the diploid somatic cells. Okay, what are somatic cells? Somatic cells are all of your other cells except for your germ cells, which are involved in formation of gametes. Every other cell undergoes cell division by mitosis itself. Very rarely haploid cells also can undergo mitosis. Example, male honeybees. Male honeybees are themselves haploid. Therefore, even in haploid cells and male honeybees, mitosis will take place, but otherwise mostly it happens in diploid cells. But in plants, it can happen both in haploid and diploid cells. Remember, we learnt about various types of plant groups, bryophytes, pteridophytes, gymnosperms, algae, angiosperms and all of that. I told you how plants have a gametophyte and a sporophytic phase. Gametophyte is haploid, sporophyte is diploid. So we see cell division happening, mitotic cell division happening in the gametophytic phase, which is haploid also. We see mitotic cell division happening in the sporophytic phase, which is diploid also. So in plants, it can happen both in haploid as well as diploid cells. And mitosis is said to be an equational division. Now I'll explain to you what that means. Let's say the parent cell that had to divide was diploid, which means let's say it had 4 chromosomes. After cell division, it will give rise to 2 daughter cells. Both of them will have the same ploidy that the parent cell had. They will also be diploid. Okay, and they will also have 4 chromosomes, very similar to the parent cell. If the parent cell had 10 chromosomes, that will also have 10 chromosomes. If that parent cell had 20 chromosomes, the daughter cells also will have 20 chromosomes. So whatever is the chromosome number here, it will be equal to the chromosome number in the daughter cells. That's why we call it equational division. Alright, now let's see what are the different stages in M phase. During M phase, there are 2 subphases. One is karyokinesis. Everybody knows karyon means nucleus, right? Prokaryote, eukaryote, karyon means nucleus. So first phase, nuclear division will happen. Why is it so important? Why can't the cell wholly just divide at once? Because DNA information is very important. It has information for pretty much every activity that takes place inside a cell, and the cell has to ensure that both the progeny cells receive equal complete set of DNA. Let's say there were 20 mitochondria present in the parent cell. During cell division of cytoplasm, one cell received 11, one cell received 9 mitochondria. That is not a problem. If there is need of mitochondria, mitochondria can, divide by fission and give rise to more mitochondria, right? Likewise, any other organelle, if there is a need, more of them can form later depending upon the need. But for the cell to function normally, it should have a complete set of genetic information, right? So therefore, that proper separation of DNA so that both the cells get complete set of genetic information is very, very crucial. So that happens first in the process called karyokinesis. The second one is called cytokinesis. If karyokinesis is nuclear division, it's very obvious that cytokinesis is basically division of cytoplasm. So it is cytoplasmic division. Okay, first over here in karyokinesis, see, this whole thing is M phase. This entirely is M phase. Here you have karyokinesis, here you have cytokinesis. Karyokinesis is further divided into 4 stages or phases, you can call it. Prophase, Metaphase, Anaphase and Telophase. We'll have to learn about what happens exactly in each of these phases. Okay? Let's start with, I've already told you there are 4 phases: prophase, metaphase, anaphase, telophase. PMAT, you remember it in the exact sequence. So after G2, which is the phase that comes, it is prophase, then followed by meta, ana, telo, and then finally cytokinesis will happen. So let's see what happens during prophase. Of course, this is the first stage of karyokinesis. How do we know that a cell has entered prophase? There will be something that will demarcate the cell from interphase and prophase. Of course, the cell does not know that it's right now in interphase, it's now in prophase. We have made all of these for our understanding. So when can we say that a cell has entered prophase? Is when you can see the DNA getting condensed. Now the DNA inside the cell does not always exist in the form of chromosomes. When the cell is not dividing, when it is in interphase, it exists in a state called chromatin. Okay, so this is our naked DNA. The DNA rarely exists as naked DNA. It will always be in some form of condensation, some level of condensation. So when it condenses, it winds around proteins and becomes more and more compact. More about this whole process you'll learn in another chapter. For now, just remember, DNA can exist in the form of chromatin or it can exist in the form of chromosome. But when the cell is not yet dividing, when it is in interphase, it will exist in the form of chromatin. Okay? But when the cell has to undergo cell division, it will become even more condensed, even more compactly coiled and form the chromosome. So when this condensation of chromatin to chromosome begins, then we say that the cell has entered karyokinesis prophase. Okay, so it is marked by condensation of DNA. It will start to condense. Okay. Yeah. Now, here you can see that the chromosomes will be composed of two chromatids because replication is already over. There will be two chromatids. There is a centromere which attaches this chromatid to this chromatid. In the last class, we've also studied how on either sides of the centromere, you will have a proteinaceous structure called kinetochore present. So that will also be present here. So this is what has happened to the DNA in prophase, at the beginning of prophase. During S phase, the centrosome had also duplicated, right? The centrosome also would have duplicated. Now this centrosome which has duplicated, this is one and let's say this is the other, they will start moving towards opposite poles. Towards the end of prophase, the centrosome would have reached the opposite poles. Remember, this is only in animal cells towards the end of prophase, or you can say late prophase. Okay, the centrosome would have reached the opposite poles of the cell. I told you that the function of the centrosome in animal cells is to help in the formation of spindle fiber. Spindle fiber is, remember I told you last class, there are cytoskeletal elements present inside of our cells. They are made up of microtubules, microfilaments and intermediate filaments. These microtubules will reorganize and form spindle fiber, which will help in separating the chromosomes between two daughter cells. Okay, so this centriole or the centrosomes that have moved to the opposite poles, they will start giving out microtubules that appears like a star and we call them aster. Okay, so it starts radiating out microtubules which we will call the asters. These asters, along with the spindle fibers that it's forming from both the poles, form what we call the mitotic apparatus. At the end of prophase, you do not see organelles like Golgi apparatus, endoplasmic reticulum anymore inside the cell. Also, the nuclear envelope would have disintegrated. You don't see a nuclear envelope right now. All of the chromosomes condensed will be present right in the cytoplasm itself. And also the nucleus will no longer be visible. That marks the end of prophase. Now, after prophase, what we have next is the metaphase. How do we know that the cell is in metaphase? Metaphase starts with M for middle. All of the chromosomes that are now fully condensed, they will start and align like one straight line that you form in your school assembly. They will come and align at the center of the cell. Okay, so all of the chromosomes that had condensed will come and align at the center of the cell. And also we know that it is metaphase when the nuclear envelope would have completely disintegrated. Okay, so now chromosomes are spread through the cytoplasm of the cell. Condensation would be complete. It started condensation in prophase. Right now it's completely maximum compact chromosomes, very short compact chromosomes would have formed. They will align themselves and arrange themselves at the center of the cell, which we call the equatorial plate. Okay, Right now, it is this phase in which if you have to study about chromosomes, you can study very well, because here their morphology, their number, all of that is very clearly visible under the microscope. Alright, at this point, you can say that each chromosome, you can clearly see two distinct chromatids that are held together at the centromere. And on either side of the centromere, you will have these kinetochores present. To the kinetochores is where the mitotic spindle will come and attach later. Okay. So, these kinetochores are the site at which spindle fiber that have started to arise from the asters radiate from the centrosomes that will come and attach at the kinetochore itself. Okay, so here you can see very well how this is the centrosome, this is the centrosome opposite pole, it's radiating out microtubules which we call the aster. It's also giving out microtubules in the form of spindle fibers. From each pole to each chromosome, spindle fibers are attached. From this pole to one kinetochore, from this pole to the other kinetochore. Okay, so at metaphase of mitosis, you can see one cell plate being formed. One cell plate will form at the center of the cell. We call this the equatorial plate or the metaphase plate. Why equatorial plate? Because since similar to how in our earth the equator imaginary line runs exactly at the center, in the cell also it almost at the center this alignment happens. Okay. Yeah, the rest of the things I've already told you. That's about metaphase. Next question. Next phase. Anaphase. What happens during anaphase? During anaphase, the centromere will split. What was the centromere doing? The centromere was keeping the two sister chromatids together. During anaphase, remember, through prophase, metaphase, anaphase, telophase, the chromosomes have to separate and the nucleus has to divide. So right now, during anaphase, that separation begins. The two sister chromatids, the copies, identical copies that were present, they will start getting separated from each other as the centromere that was holding them both together will split. Okay. So during anaphase, you can see that the centromere will split. It's not like for one chromosome it's splitting at one point and 3 minutes later it's splitting for the other chromosome. How many other chromosomes were present in the cell in metaphase, and all of those chromosomes simultaneously, the cell in a very coordinated manner will split the centromere. Okay, when the centromere splits, you have chromatids belonging to a same chromosome are called sister chromatids. So they were sister chromatids up until this point because they were connected. Now that they are separated, you no longer call them sister chromatids, you can call them sister chromosomes, right? So these individual chromosomes will start being pulled on towards the opposite poles. So these microtubules that are forming the spindle fibers, they will become depolymerized, become shorter and shorter and shorter, and these sister chromosomes will start moving towards the opposite poles. The two daughter chromatids are now referred to as daughter chromosomes of the future nuclei. They will begin to migrate towards the two opposite poles. I've told you that. As each chromosome moves away from the equatorial plate, yeah, this is important to note. So, this is the centromere. Here is the kinetochore. To the kinetochore, this spindle fiber is attached. Since the point of attachment is the kinetochore, which is present attached to the centromere, this will be towards the pole leading, and trailing behind you will have the arms of chromosomes. Okay, so this will have, centromere will have the leading edge. You can see both sides, wherever it is attached, there it is towards, it's being pulled this way. So there it will be towards the centromere will be in front towards the pole leading, and the two arms will trail behind. It's very similar to how if you tie a thread with another thread in the center and pull it, the point of attachment will be in the front and the other two parts will trail behind, right? Very similarly, even over here, the centromere will have the leading edge and the arms will trail behind. Okay? Last phase of karyokinesis, it's called the telophase. During telophase, the events exactly happens opposite of prophase. During prophase, what happened? DNA, chromatin condensed to form chromosomes. Now chromosomes decondensed to form chromatin. Nuclear envelope disappeared. Now nuclear envelope will reappear. Nucleolus disappeared, nucleolus will reappear. Organelles like ER and Golgi disappeared, now organelles like ER and Golgi will reappear. Okay, so that's exactly what happens. This is the final stage of karyokinesis. The nucleus is almost going to divide right now. The chromosomes are separated. It's just that around this genetic material a membrane has to form, right? So the chromosomes that have reached their respective poles decondense and become chromatin. The individual chromosomes, you cannot identify individual chromosomes like you used to in metaphase anymore. Okay, so they will lose their individuality because they are all decondensing and becoming thread-like. The individual chromosomes can no longer be seen and each set of chromatin tends to collect at each poles. Nuclear envelope will reappear. Nucleolus, Golgi and ER also will reform. So at the end of telophase, the nuclear division has been completed. We are done with karyokinesis. What do we have next? What we have next is called cytokinesis. Cytokinesis is basically the division of the cytoplasm, right? Cytokinesis is cytoplasmic division. This happens differently in plants and in animals. First, let me show you how it happens in plants. Let's consider a cell in which nuclear division has already occurred. So here you have a plant cell. The nuclear envelope would have reformed. So you have proper nuclei. Separated here. Within that the DNA would have decondensed. So chromosomes do not have clear identity. DNA has decondensed, chromosome have decondensed. Right now in between these two a cell wall has to form. Remember, in plant cells cell wall is there, in animal cells cell wall is not there. So in plant cells cell plate will start getting laid. Vesicles from Golgi apparatus will start laying the cell wall from the center towards the periphery like this. The vesicles will have cell wall material. It will start depositing the cell wall material starting from the center towards the periphery. What forms here is called a cell plate. So during cytokinesis in plants, a cell plate forms. So it happens, remember, from the center towards the periphery. What happens in animals? In animals, the direction is reverse. Remember, animals lack a cell wall. Let's consider an animal cell. Okay. So let's say in this animal cell, the nucleus is duplicated. Karyokinesis is done and dusted. Cytokinesis has to take place. Okay, so the DNA here also decondensed. Over here, outermost layer is the cell membrane. The cell membrane kinds of pinches off from the periphery, forms a furrow-like thing, similar to how you take out chapati dough from chapati to roll your chapatis, right? So like that, it will form a furrow. And this furrow will become deeper and deeper from both the sides, eventually meeting at one place, and that will separate both of these cells together and you will get two separate cells, two complete cells with its own nucleus each. Okay, so in animals it happens from the periphery to the center, formation of furrow, and in plants the cell plate is laid from the center towards the periphery by means of vesicles that are formed from Golgi apparatus. Okay, so that is cytokinesis for you. Now let's talk about the significance of mitosis. One significance I already explained at the beginning of the class itself. If a unicellular organism has to grow and become multicellular, then obviously in growth, cell division plays a very important role. Mitosis plays a very important role. In plants, all cells don't have the ability to undergo cell division. Only specific types of cells can undergo cell division. We've learnt about it in the chapter Anatomy of Flowering Plants. We call such tissues meristematic tissues. Permanent tissues would have lost that ability. Basically, what mitosis does is, it will restore the nucleocytoplasmic ratio, which would have gotten disturbed. Another thing that it helps in is repair. If there is any damage in your epithelial lining or any part of your body, some tissue or cells, the barrier is broken. In order to repair it, cells present will start dividing until it is repaired. So mitosis happens and it helps in growth. It restores the nuclear cytoplasmic ratio and it also helps in repair. Okay, generally the cells that are exposed to the outer environment or exposed to different things inside, like if you eat food, your epithelial lining of your gut, it's exposed to a lot of things, right? Because we eat a lot of things every day. So such things are prone to damage. Over there, mitosis happens very frequently to repair the damage. Cells of the upper layer of epidermis, cells of the lining of the gut and blood cells are being constantly replaced. Okay, so that is the function of mitosis. Now we have to learn about what happens during meiosis, the second type of division. Well, unlike mitosis that we learnt about previously, this is not an equational division. This is reductional division. What does that mean? Reductional division means if a parent cell has two sets of chromosomes and is diploid, let's say it has 4 chromosomes, then this parent cell will give rise to 4 daughter cells. Each of these daughter cells will have half the number of chromosomes and will be haploid, and they will have 2 chromosomes each. Okay? Since the number of chromosomes in the parent cell is reduced to half in the daughter cell, you call it reductional division. When does this kind of cell division happen? Previously, we learned that mitosis happens in somatic cells during growth and repair. When does meiosis happen? Meiosis happens in generally sexually reproducing organisms during the formation of gametes. Let's take humans for example. The process of gamete formation, we call it gametogenesis. When in males gametes are formed, sperms are formed, and in females the egg cell is formed. Both the sperm cell and the egg cell, in general, gametes are haploid, right? So during the formation of haploid, a diploid cell will undergo meiosis and form the haploid gametes. If you're wondering why should another type of cell division even exist, why can't gametes just form by mitosis? What happens if the gametes are also diploid? If the gametes are diploid, then the number of chromosomes in a population cannot be maintained. Now, if you consider us humans, all normal humans will have 46 chromosomes in our somatic cells, right? Sometimes due to some genetic disorders, there may be 47 chromosomes, 45 chromosomes, but that's not something that normally happens. Same way, even other organisms, if you consider one species, the number of chromosomes is fixed in healthy individuals. That will vary if meiosis does not take place during gamete formation. How would that vary? Let's understand. Let's say in humans, there is a gamete mother cell which is diploid. If it undergoes meiosis, it will produce haploid cell, sperm. Gamete mother cell in female undergoes meiosis, produces haploid cell. So ultimately our zygote will be diploid. This will undergo mitosis and the diploid condition will be maintained. What would happen if gamete formation happened via mitosis? Gamete mother cells are diploid. Gametes also will be diploid. Imagine if a sperm containing 46 chromosomes fuses with an egg which also contains 46 chromosomes, then the zygote is going to have 92 chromosomes. It's going to be tetraploid. Now, if this zygote develops and reaches maturity and if it has to reproduce, then the gametes that this zygote or this individual that has formed from this zygote will have 92 chromosomes, and the next generation would be 184 chromosomes in the zygote, right? That doesn't happen. In fact, if more than the fixed number of chromosomes, the normal number of chromosomes only, the zygote or the embryo will not be viable. So this doesn't happen in nature. In nature, we see for a species, the number of chromosomes is fixed in normal individuals, right? So the reason is because of meiosis. So what happens during meiosis? The number of chromosomes is reduced. So a haploid phase you see. How is that haploid phase restored to diploid phase? By the process of fertilization. Because of meiosis, we get haploid gametes. Because of fertilization, diploid condition is restored in the zygote. Okay? So that's the reason why in a population belonging to one species, all individuals will have the same number of chromosomes normally. Okay? In normal healthy individuals, the number of chromosomes will be the same. Right? Now, what are the different stages in meiosis? Mitosis was simple. You had karyokinesis, cytokinesis, and then in karyokinesis, you had prophase, metaphase, anaphase, telophase. During meiosis, remember, reduction in the number of chromosomes need to take place. So, meiosis happens in two sequential division cycles that we call meiosis I and meiosis II. In meiosis I, you have karyokinesis, cytokinesis. In karyokinesis of meiosis I, you have prophase I, metaphase I, anaphase I, telophase I. Again, you have meiosis II where you have prophase II, metaphase II, anaphase II, telophase II. So, it's like it happens twice. Okay. So, in meiosis, there are two sequential cycles which we call meiosis I and meiosis II. Though meiosis happens in two sequential stages, there's no different interphase for meiosis I and different interphase for meiosis II. If you look at the cell cycle of a germ cell which undergoes meiosis, you can see how you'll have the G1 phase, you'll have the S phase, you'll have the G2 phase, and within this M phase itself, you'll have meiosis I and meiosis II. In between meiosis I and meiosis II, there is no interphase again. Okay, just one cycle of DNA replication happens during this interphase here. In between these two, there is no DNA replication again because that will not serve the purpose. The whole reason why we are doing this twice is because you want to reduce the number of chromosomes. If DNA replication happens there, what is the point? It cannot get reduced, right? Yeah, so there will be a single cycle of DNA replication. Meiosis is very, very essential because that is what brings about variation in a population. How exactly it happens, I'll show you as we move on. So for now, understand that in a population, you see variation with respect to so many characters. Like it could be the color of hair. Some people have brown, some people have black, some people have red, some people are blonde. So you have different colors. The texture of hair is variable. Some people have it curly, some people have it straight, some people are wavy, the height differs, the complexion differs. A lot of variations you see in nature. And that is because of the process of meiosis. One of the reasons why you see variation is meiosis. So at the end of meiosis, you get 4 haploid cells. At the end of mitosis, we got 2 diploid cells. That is another difference. So we'll first learn what happens in meiosis I and then we'll learn what happens in meiosis II. Meiosis I has prophase I, metaphase I, anaphase I, telophase I. Okay, so before that, I'll give you an overview of what happens during meiosis. I'm saying that reduction in the number of chromosomes will happen. Diploid cell will give rise to 4 haploid cells. What exactly is happening with respect to the number of chromosomes in different phases, we'll understand. About 2C condition, 4C condition, I've explained earlier only in S phase, right? So keep that in mind and pay close attention to what I'm about to say now. We'll start off from G1 phase. At G1 phase, I want you to notice, don't look at everything else that is there on the board, just notice on whatever I'm pointing at, okay? Yeah. So let's take a look at this cell right here. It is a brand new cell that has just now formed. It has 4 chromosomes. It is diploid in nature. It has 4 chromosomes. Each chromosome has 1 chromatid. So this will be in 2C condition right now. It has just now formed. Now what is going to happen? It's going to enter interphase. Within interphase, it's going to enter the G1 phase. During G1 phase, is any change going to happen with respect to DNA? No change will happen. DNA replication will not take place. The cell will just increase in size. So the small cell that was here will increase in size here. The number of chromosomes, the number of chromatids, the ploidy, there is no change. Even after G1 phase, the cell will still have 4 chromosomes. It will still be diploid. It will still have 2C condition. Right? What is going to happen during S phase? During S phase, DNA is going to make an extra copy. When it makes an extra copy, the chromosomes will now have 2 chromatids each. Right? So after S phase, you can see towards the end of S phase, after replication has taken place, there are 4 chromosomes similar to how it was earlier. But look at the difference in the number of chromatids. Now that each of these chromatids have made an extra copy for themselves during S phase, you can see here after end phase, towards the end of S phase, after replication is complete, that each chromosome will contain 2 chromatids. If it contains 2 chromatids, then we call it the 4C condition. It's not because it contains 2 chromatids you call it 4C condition. Basically, the DNA content has doubled. That is why we call it the 4C condition. So right now, since there is twice the amount of DNA here, you call it the 4C condition. Okay, where is this? This is in S phase. Now what happens? The cell will move to G2 phase. During G2 phase, is there any change in the number of chromosomes or chromatids? No change absolutely. However, it was during S phase, it will be exactly the same. It will contain 4 chromosomes. It will still be diploid, but it will have 4C condition. So during G1, it is 2C condition. After S phase and in G2 phase, it will be 4C condition. Now pay close attention to what is going to happen during meiosis I. During meiosis I, which you can see here, this is meiosis I, the first division. First the cell will divide and give rise to 2 cells. Those 2 cells will further divide to give rise to 4 cells. That is what happens. So during meiosis, one parent cell will divide to give rise to 2 cells. I want you to notice how after meiosis I, here is meiosis I, this division is meiosis I. I want you to notice how after meiosis I, the number of chromosomes have reduced. Initially, there were 4 chromosomes. Right now, there are 2 chromosomes. But have the number of chromatids reduced? The number of chromatids have not reduced. Is this cell haploid or diploid now? It has become haploid because the number of chromosomes have reduced. How did I tell you we count the chromosomes? We count the chromosomes based on how many centromeres are present. Here you have 1 centromere, 2, 3 and 4. So you have 4 chromosomes. Right now, the chromosomes have separated into 2 daughter cells. The chromosomes have reduced in number such that each daughter cell has only 2 chromosomes. So from 4 chromosomes, you have 2 chromosomes each cell after meiosis I. If the number of chromosomes have reduced, then we say that the cell has become diploid. It was 4 here, it's 2 here, it's reduced to half. So the cell, it has become haploid, not diploid. Okay. So at this point, the daughter cells will have 2 chromosomes with 2 chromatids. What is the condition here? If you compare the DNA content with this and this, has in one cell here and one cell here, is the DNA content the same or it is reduced? The DNA content is reduced to half. If here it was 4C, how much should it be here? It should be 2C here and it is 2C. Some of them might ask, but there are 2 chromatids here, how is it 2C? It does not depend on how many chromatids are present. 2C and 4C basically denotes the amount of DNA there is in a cell. Okay, so here, how much ever DNA was there in 2C, in this cell, I want you to compare this cell here and this cell here and tell me, is there any difference in the amount of DNA? The amount of DNA is the same here. How much amount of DNA is present is the same. Over here, you can think of it like you had one pizza and you cut it into 4 parts and you had 4 parts. Over here, you can think that you had one pizza and you cut it into half and you have it in 2 parts. That's the only difference, right? But the content, the amount of DNA is the same here and here. So this is also 2C condition. What is going to happen next? After meiosis II, each of these cells are going to divide in a fashion which is very similar to what happens during mitosis. What happens during mitosis? The centromere will split, the sister chromatids are pulled towards the opposite poles and 2 new cells are formed. Those 2 new cells will have only one chromatid in the chromosome. So after this, when meiosis II happens, the centromere will split such that each daughter cell that is formed after meiosis II will get only chromosomes with 1 chromatid. Now compare this and this. Is there a change in the number of chromosomes? There is no change in the number of chromosomes. There are 2 chromosomes here. There are 2 chromosomes here. Is there a change in the number of chromatids? Yes. Here each chromosome had 2 chromatids. And here each chromosome has 1 chromatid. Look at this cell and this cell. Is there change in the content of DNA or amount of DNA? Yes. how much ever DNA was present here, that is reduced to half here. So if it was in 2C condition at after M1 phase, it will become even reduced to 1C condition after M2 or meiosis II. Okay, I hope this is clear. So this is just an overview. You will understand it better as we learn in detail how this type of cell division happens in meiosis. Okay, let's start with prophase 1 of meiosis I. This is relatively longer compared to the prophase of mitosis. Remember, I told you because of meiosis we get two very important things, or there are two important significance of meiosis. Firstly, it maintains a uniform chromosome number in the entire population. Secondly, it brings about variation. I told you how we all vary and differ from each other with respect to so many characters, and one of the reasons why it happens is meiosis, right? So it is exactly in this very phase of meiosis, in prophase 1, that variations are brought about. How exactly it happens, I'll tell you. Now within prophase 1 of meiosis, you have 5 substages, which are leptotene, zygotene, pachytene, diplotene and diakinesis. Leptotene, zygotene, pachytene, diplotene and diakinesis. Okay, so these are the 5 substages in prophase 1. Now, before we start understanding how variations are brought about, I want to teach you some basic things about our chromosomes that you need to understand how variations occur. Okay, let's take one example. We all are diploid individuals, which means we have 2 sets of chromosomes, right? All diploid organisms have 2 sets of chromosomes. How do they get 2 sets of chromosomes? One set of chromosome they receive from the mother through her egg. The other set of chromosome they receive through the father through his sperm. So when fertilization happens during sexual reproduction, from the male parent, from the female parent, you get 1 set of chromosomes each, and we diploid organisms will have 2 sets of chromosomes. Okay, so here for example, I have taken 2 sets of chromosomes. This is one. This is the first set of chromosome that I have received from my mother. This is the second set of chromosome that I have received from the father. In order to distinguish between which is mother, which is father's chromosome, I have used different colors. Pink for girls, blue for boys. So pink is the chromosome that I've received from the mother. Blue are the chromosome that I've received from my father. So there will be one of my mother's chromosome that pairs or matches exactly with one of my father's chromosome. Okay, so if you look at these chromosomes here, this and this is exactly the same in the sense, if in this chromosome at this position there is information for hair color, in this chromosome also there will be information for hair color. If at this position in this chromosome there is information for eye color, in this chromosome also at this position there will be information for eye color. Okay? Whenever such chromosomes are there where information for the same things is present at the same location in 2 chromosomes belonging to 2 different parents from 2 different sets, we call such chromosomes as homologous chromosomes. This is the maternal set, this is the paternal set. This chromosome from the maternal set is homologous with this chromosome of the paternal set. Wherever in this chromosome information is there for a particular character at this position, this chromosome will have the information for the same. This and this cannot be homologous because this contains different set of genes, this contains different set of genes. This and this is homologous. I hope you're getting my point. Let's take an example. Mother has brown hair that is curly. Father has red hair that is straight. Okay, we'll consider these 2 characters. Mother's hair is brown and curly. So at this position here, you have information for hair color. Mother's hair is brown, right? So for this, you have brown hair color information present in the mother's chromosome as she has brown hair. At the same location, you have the same information for the father. At the same position, there is information for red hair color. Okay? The character is the same. Next, at this location, which is marked in yellow color, there is information for hair texture. Mother's hair, as you can see clearly, is curly. So at this position in mother's chromosome, there is information for curly hair. But in father's chromosome, since he has straight hair, there is information for straight hair. Such chromosomes which have information for the same character at the same location are called homologous chromosomes. That is one word that you've understood now. You also have to understand what sister chromatids and non-sister chromatids mean. Two chromatids belonging to the same chromosome, these two belonging to the same chromosome are called sister chromatids. Now, this chromatid from this chromosome and another chromatid from another chromosome which is homologous to it are called non-sister chromatids. Okay? These are sister chromatids, this and this is sister chromatid, this and this is non-sister. This and this is nothing because they are not even homologous chromosomes. Okay, sister and non-sister, especially non-sister chromatids, they are chromatids belonging to two different chromosomes which are homologous. Okay, I will be using these terms going forward, so pay attention. Alright, now let's start with leptotene. Remember, leptotene, zygotene, pachytene, diplotene, diakinesis is essentially different stages in prophase I of meiosis. Whatever events happen during prophase of mitosis, the same things are going to happen even here. Nuclear envelope is going to disappear. DNA condensation will begin. Nucleolus, ER, Golgi, all will disappear by the end of prophase. Okay, so it's starting here. Along with these changes, variations also, cause for variations also will happen here. Okay, so firstly, during leptotene, this is not very interesting, nothing interesting happens here. During leptotene, what happens is chromosomes will start appearing under the microscope because DNA condensation is happening. So throughout the course of leptotene, compaction of chromosomes, DNA becoming more and more condensed, shorter and thicker, that will continue here. Let's talk about the next phase, zygotene. During zygotene, which is the second stage of prophase, homologous chromosomes will pair opposite each other. Okay? So if this was one chromosome from the mother and this is another chromosome from the father, they both will join like this, attached to each other like this. They will come in pair. Which chromosomes will pair? Specifically, homologous chromosomes will come in pair like that. That process is called synapsis. These homologous chromosomes are held together by what we call a synaptonemal complex. That is what glues these two homologous chromosomes together. It acts like a glue that keeps these two together, or basically this whole complex is called the synaptonemal complex. Okay. These paired chromosomes are homologous chromosomes. This is formed by a complex structure. Okay, that complex structure that forms with the glue holding the two homologous chromosomes together, that is your synaptonemal complex. At this point, if you look at this chromosome, you can see that two chromosomes are attached to each other. So we call this paired homologous chromosome as the bivalent. Bi means two, right? So bivalent is the two homologous chromosomes together. If each homologous chromosome at this point has two chromatids, then there is a total of 4 chromatids. So you can even call it a tetrad. Remember, prophase is still proceeding. So condensation is still happening. Okay, so as condensation of DNA is happening, these homologous chromosomes have paired. Next, pachytene. So here in a bivalent you will have 4 chromatids. In a tetrad or a bivalent you will have 4 chromatids. since condensation is still happening, you can see them being more compactly arranged. They will become more distinct and clearly visible as tetrads. At this point, something called recombination will take place, or a process called crossing over will take place. I'll tell you what happens. Look here. Let's take these two chromosomes for example. These both are homologous chromosomes. In leptoteen, what happened? They both had come together. Assume they both are joined to each other like this. At certain locations, exchange of genetic material will happen between these two homologous chromosomes. At random locations, exchange will happen such that exchange is happening between non-sister chromatids, such that we can say this portion and this portion will get exchanged in such a way that in one chromosome is like this, the other chromosome will be like this. Okay? So here, this female parent initially had curly hair gene here, gene for curly hair here, and now that exchange has happened, the gene that the father had for straight hair will be here. So you got a new combination. Earlier, curly hair was with brown color, straight hair was with red color. Right now, since there is exchange of segment happening here, you will have a new combination of brown hair that is curly. Brown hair that is straight. Initially, you had brown hair curly, red hair straight. After this crossing over happens, you will get newer combinations that will bring about variation in a population. Are you getting my point? And here at this point, you had red hair that was straight. Right now, you have a new combination of red hair that is curly. Okay, I'm showing this to you separately as two different chromosomes, but the chromosomes have not yet separated. While they are still attached, while they are still paired, while they are still synapsed, this crossing over or exchange will take place. Because of this crossing over or exchange, newer variations, newer recombinations you can see in a population. Why should there be exchange between non-sister chromatids? What will happen if there is exchange between sister chromatids? There won't be any change at all, because sister chromatids are identical copies of each other. Initially, there was just this one copy, right? Initially, there was just this one copy. Now, during S phase, an identical copy, exact replica was made. So, whatever is here is here. Even if there is exchanged, there will be nothing changed because they both themselves are the same, right? Therefore, it is pointless for exchange to happen between sister chromatids. So, exchange of genetic material or segments of DNA will happen between non-sister chromatids of homologous chromosomes. So, that will happen during pachytene. So, wherever crossing over has happened, over there you can see the formation of nodules, which we call recombination nodules. If the entire segment only gets changed, if this entirely gets changed, if this entirely goes here, this entirely goes here, that also will not serve any purpose, because brown is always going to be with curly, red is always going to be with straight. Newer combinations can happen when only specific random segments can get exchanged. That is what will happen during crossing over. That is why you see variation in the population. Alright. So, yeah. So, this crossing over is basically exchange of genetic material. An enzyme catalyzes this process, and that enzyme is called recombinase. Okay. That is what brings about genetic recombination. Now, let's talk about the next phase called diplotene. During diplotene, what happens is, right now crossing over is over. Recombination nodules will still be present at the sites of crossing over, just at those locations where crossing over has taken place. Let's assume at my joints here the crossing over has happened. Everywhere else, the synaptonemal complex will dissolve except for wherever crossing over has happened. Like right now, crossing over has happened at my joints, except for that everywhere else it has dissolved and the two homologous chromosomes have kind of separated, right? Now, because of the attachment of chromosomes to each other at the site of crossing over, you can see X-shaped structures forming. It forms X-shaped structures, right? These are called chiasmata. X-shaped structures are formed. They are called chiasmata. You see them during diplotene. So, at the beginning of diplotene, they will start separating and they will be held together only at the regions of crossing over. There you find X-shaped structures, X-like structures, because they are attached like this still. That is called chiasmata. Now, you may have learnt in the chapter human reproduction how females in many vertebrates, they start producing or they start the process of meiosis, production of egg, oogenesis process even before the baby is born. While the baby is still in the mother of the womb, only oogenesis would have started, right? But it will not complete. Actual egg formation release will happen only around the time of puberty, during menarche. Before that, it would have stopped at one point. It would have stopped at this very phase, which is diplotene. So the oocytes, which are cells which will undergo meiosis to give rise to the egg cell, in several vertebrates will be stalled in the diplotene stage for several years and months. Okay, so that is one point you should remember. Lastly, diakinesis. This is the last stage. Throughout this, from prophase condensation of DNA started to diakinesis, DNA condensation will continue. At diakinesis, DNA is almost fully condensed into chromosomes. It's still showing condensation. Chromosomes were held together at the points of chiasmata. Now that the chromosomes are becoming shorter and shorter and shorter because of condensation, it will seem like this point of attachment is moving farther and farther and farther away towards the tip. So we call that terminalization of chiasmata. Terminal means end. Chiasmata is moving towards the tip. It's not really moving, but it appears like it's moving because the chromosome is becoming shorter and shorter. It will seem like this point of joint, where it was joined, it is moving towards the tip. It will seem like that. Okay. So by the end of diakinesis, chromosomes are super condensed, fully condensed, and mitotic spindle also will appear because it's the end of prophase. Nuclear envelope, nucleolus, Golgi, all of them will disappear. Now it will transition into metaphase 1 of meiosis. During metaphase 1 of meiosis, something different from metaphase of mitosis happens. In metaphase of mitosis, you had just one metaphasic plate or equatorial plate. Right now, you will have two. These two are homologous chromosomes, these two are homologous chromosomes. Okay, crossing over has happened. Earlier, we saw that one chromosome had only maternal genes, the other chromosome had only paternal genes. Now, after crossing over, both chromosomes are a mixture of both maternal and paternal genes. They will come and form, homologous chromosomes will form two metaphase plates during meiosis 1. And again, they will attach at the kinetochore of the chromosomes. Over here, I want you to notice how centromere splitting does not happen during anaphase. Okay, homologous chromosomes themselves will be pulled towards opposite poles. This is metaphase. At the kinetochore, spindle fibers are arranged, attached. During anaphase, entire homologous chromosomes are pulled apart. We saw in mitosis how the centromere split and the chromatids, sister chromatids, were pulled apart. Over here, homologous chromosomes itself is pulled apart. And then you have telophase 2, where, similar to how it happened in mitosis, DNA will decondense, not fully decondense, because again meiosis 2 has to happen. To a small extent, it will decondense, nuclear envelope will reappear, other organelles will reappear, all of that will happen during telophase. I want you to notice how the number of chromosomes have reduced. Initially, we started off with 4. There's 4 here, crossing over happened, metaphase happened. I want you to notice how in these daughter cells that you got after meiosis 1, there are 2 cells in each chromosome. So the reduction in the number of chromosomes has already happened. It's already a haploid cell. But further reduction to 1C condition will happen after meiosis 2. What happens during meiosis 2? Metaphase 1, whatever happens, I've listed the details here. Align on the equatorial plate, kinetochore, they are connected, homologous chromosomes separate during anaphase. This is for your notes, I've already explained it. During telophase, two cells will be formed, nuclear membrane, nucleolus, all of that will reappear, but it will not fully decondense, okay. So after telophase, before meiosis II, after telophase 1 and before meiosis II, there is a very brief period called interkinesis. During interkinesis, like interphase, DNA replication will not happen. It's a small brief resting phase between the two stages or the two cycles of meiosis. But over here, there is no G1, S, G2. Interkinesis is different, interphase is different. Okay. So this is interkinesis. This is the stage between two meiotic divisions. It's a very brief period where DNA replication will not happen. After this, you have meiosis II. Meiosis II is exactly like mitosis. Okay, whatever happened in mitosis, the same thing will happen even in meiosis II. After meiosis I, we had two cells with two chromosomes each, two chromatids each. During prophase, condensation will happen, spindle assembly will happen. During metaphase, one metaphasic plate or equatorial plate will form in each cell. During anaphase, the centromere will split and pull the sister chromatids towards the opposite pole. At telophase, decondensation of chromosome, reappearance of nuclear envelope, nucleolus, ER and Golgi, all of that will happen exactly like mitosis. So at the end of meiosis II, you will have a total of 4 cells. Each of them is haploid. Each of them has 2 chromosomes. Each of them is in 1C condition. Alright, so that's the end of meiosis. What is the significance of meiosis? 2 very important things. One thing, it conserves the chromosome number in the individuals of a species. Secondly, it brings about variations in a population. I have homework for you. In the comments, after you watch this video, if you've understood everything, you have to list the differences between mitosis and meiosis in the comment section. I will check it. Okay. So the significance of meiosis, 2 important points. Chromosome number is maintained and also it causes genetic variation in a population. I hope this session was helpful. If you have any doubts at all, please feel free to drop your doubts in the comments. I will 100% answer them as soon as I can. I'll see you in the next class. Until then, take care, keep studying hard. All the very best. Thank you.